Hello and welcome everybody to the Central and South Genomics Year in Review. It is lovely to see all of you who have joined us so far. We've got a lot to go through, so I'm not going to do the kind of we'll give it a minute. We will get ourselves moving today, and as others join, they can catch up with where we've got to. My name is Vicky Marshall. If you don't know me, I work as the Managing Director of the Central and South Genomic Medicine Service Alliance, and I'm just going to get us kicked off today. So as not to eat into the time for the actual presentations, I wanted to say, really, this is an opportunity to have just an hour to have a look back at what we've delivered this year and a little bit of thinking as to what's to come in 2025. I hope you've managed to get a cup of tea. It's supposed to be quite a low-key hour and something that hopefully is of interest. It will lead into a little document that we issue this year as a review of the year. And I would say if there's anything you've worked on, written up, or your team have been involved in that you'd be keen for us to include, do let us know and we can make sure that that's covered in there, either by dropping Isabel or I a line, and we'll make sure that email addresses are in the chat box as well. So I will finish off by just saying, because this is the last time you'll hear from me today, to say thank you very much for all the support those on the call have given us this year, all the work you've done with us this year, that we're very much looking forward to working with you next year. And if we didn't do much with you this year, then hopefully there's some opportunities to work together in 2025. So I will hand over now to Jenny Bell, one of the two operational directors of the Genomic Laboratory Hub in Central and South, and really hope you enjoy the next hour.
Thank you very much, Vicky, for that lovely introduction. And as we said, we're just wanting to reflect very briefly on the year that's coming to an end quite soon. I think all of us have said that it's been an exceptionally challenging and busy year. And we're going to end the session, as Vicky said, with a quick forward look. But from the Genomic Laboratory Hub side, we felt it was really important to just reflect on some of the amazing things that have been going on. And we know we can't cover everything in one single slide, but this is just a taster of some of the things we felt have really impacted over the year. So the laboratory team, supported by our clinicians, have undertaken a massive and sustained reduction in our backlog. We started the year in a not too brilliant place, and we have been able to reduce that down dramatically. That's been driven by a multitude of different things: process improvement, super session overtime, but fundamentally the dedication and commitment of all of the teams in the GLH. We've been hitting major milestones as we've gone along, with 10,000 familial hypercholesterolemia proband reports going out, 10,000 rare disease whole genome sequencing reports for probands being released, more than 12,000 large cancer panels reported, over 900 R21 cases reported, more than 15,000 NIPT sample tests. That milestone was hit this year, and our plater has processed now 3,500 samples per month consistently. That's huge. That's an enormous achievement. We've also, on the back of the delivery of the business as usual, developed amazing new services for our patients and introduced new technologies. We've got our pan-hemato-oncology panel with distributed analysis working increasingly more smoothly, and the Oxford team have been able to get their NovaSeq X machine up and running and networked, and that's fabulous. We've done exceptional amounts of training and education, supporting our trainees at all levels, both scientists and technologists. We have a broad offering of CPD that's, I'm hoping, openly accessible to all of our GLH teams. And we're developing the Genomics Training Academy, and I feel Central and South really leading the way there with our practice educators and the delivery now of virtual reality style training. We've also had the excitement of appointing GLH leads for our bioinformatics and quality. So it's been a fantastic year, full of exceptional challenge, but exceptional hard work. So to follow on the baton from Vicky, a massive thank you from the senior team in the GLH to everybody for their massive contribution. It's been a hard year, but also, if you look back, a great year. So I'll stop there and pass on to the next.
Hello, everyone. Thank you, Jenny. So my name's Karen Creed, and I'm lead midwife in Central and South GMSA, and I'm going to give you an update on the Generation study. So you may have seen some of the news articles when it was launched nationally at the beginning of October, but of course, in reality, it started way before then. National recruitment started in March 24, and many of us have been working on it much longer than that. So I'm just going to give you an overview of what's happening in Central and South, where we are now and where we're going in the future. Before I do that, though, I've just got one slide summarising what the Generation study is, because I don't want to assume that you've all heard of it. So it's a research study. It's led by Genomics England in partnership with NHS England. And what we're aiming to do is evaluate the utility and feasibility of using whole genome sequencing as a screening test for 100,000 newborn babies to identify rare genetic childhood onset conditions before the onset of symptoms. We're looking for over 200 different conditions, all of which have a treatment pathway available on the NHS and where that treatment is known to make a difference to the outcome for those babies. We estimate that we'll have about 1,000 condition suspected results during the course of the study, and those babies will go on to have confirmatory testing where we confirm that condition, we will start treatment. In addition to that, we'll be storing the data in the National Genomic Research Library to enable further research going forwards. And finally, that data is going to be stored until that child reaches the age of 16, at which point they'll be approached to ask for their consent to remain in the study. So we're also looking at the longer term implications of storing data for that child during the course of their life. Next slide, please. So, thank you. Yeah, the Generation study went live in the Southwest in March. That's when they first started recruiting. And since then, the number of sites have been increasing month on month. The blue dots on this map show the live sites, and the pink dots have either just gone live, because this slide's a week out of date, and a week is a long time in the Generation study, or they're about to go live. So it gives you an idea of how many sites we've got up and running at the moment. Yeah, in fact, we'll go on to the next slide. Thank you. So there are currently 22 sites live at 15 trusts across England this week, and 3 of those trusts are in Central and South. So we're recruiting out of 6 sites. We're at Birmingham Women's, we're at University Hospitals Birmingham, recruiting out of Heartlands, Good Hope and Solihull, and we have just gone live at Oxford, recruiting out of John Radcliffe and Horton. So doing incredibly well, and we're also in preliminary discussions with 2 new sites, Milton Keynes and Southampton. I'm just waiting to hear if they want to participate, and then we'll be working with them to support implementation going forward. Next slide, please. Thank you. So where are we in terms of recruitment? Well, nationally, we're just over 3%. This is the latest data as of 2 days ago. So just under 3% of our target. So slow start, but getting there. And nearly a third of those recruits are from Central and South. I'd just like to take the opportunity at the moment to give a shout out to our sites, because it is their determination, passion and commitment to the study from the staff there that's really driven this forward in Central and South. And you can see that we're doing very well. Birmingham Women's are the leading recruiting site in the country, doing really well. So well done everyone there. University Hospitals Birmingham started a couple of months after Birmingham Women's, but also doing well. But you can see it's more than a story of who's being recruited. We're also considering who are declining to take part, and that's part of our work going forward, to try and understand why parents perhaps don't want to participate. And I've just put some of the decline data there, so you can see that about just over a third in Birmingham Women's are saying no, and almost 2/3 in University Hospitals Birmingham. Oxford went live 2 weeks ago, and what we typically find is that it's a very slow start to recruitment in the first month, and that's rightly so, because we need to make sure that all the processes are in place and working well, but everything's going smoothly there, and we have got our first 6 recruits so far. So next slide, please. Just a little bit on samples. So we're aiming to take a cord blood sample from babies, and so far nationally we've got just over 1,500, and again almost a third are from Central and South. We also monitor where we miss those cord samples, because if we do miss a sample, it's usually due to an emergency at the point of birth, and of course the priority of mum and baby's health that mum and baby's health is a priority at that point. But there is the opportunity to take a heel prick sample. But what we find is that sometimes in those circumstances, parents decline to take part and therefore result in withdrawing from the study. So we do monitor just to make sure there's a good reason why cord samples were missed, and also just following up with heel prick tests. Next slide. Thank you. So a little bit on results. So as a GMSA, we are responsible for coordinating the results in our region. And what most GMSAs are doing are having one regional results coordinator for their entire geography. We're taking a very different approach though in our region. And that's partly because we have quite a complex geography and we have the largest population of all the GMSAs. So what we're doing is hosting a regional results coordinator within the research team in each of our 3 areas. And we've done that already in West Midlands and Thames Valley. The idea being that our results coordinator is embedded within the research team, so they're enmeshed within the study, have a really good understanding of the study. And when, of course, we don't have results to return, then they can support recruitment at that site. And then we have support from the rest of the research team to support coordinating results in the absence of our regional results coordinators. We've also funded a linked genetic counsellor in these regions, half a day a week, and their role is to support the regional results coordinator with genomics knowledge to help signpost those families for genetic counselling if and when appropriate, and also to assess the impact of the study on genetic counselling services. So we have 2 named coordinators in both West Midlands, or 2 named genetic counsellors in both West Midlands and Thames Valley, and we aim to replicate that down south in Wessex. Someone's got their hand up, I don't know. I'll carry on, and if there's a question at the end, then I will, yeah. Okay, also just want to say that a lot of work has gone into pathway mapping to make sure that we have clinicians ready to receive any condition suspected results babies, and a big shout out to the clinical teams in West Midlands and Thames Valley just for mapping all those pathways. A lot of work has gone into that, because of course we have to have those pathways in place before we started recruiting anyone to the study. Next slide, please. So where are we with results? Well, currently turnaround time is about 12 weeks, and we're aiming to reduce that to 2 weeks by the end of the study. So far, just under 500 results have been returned nationally, and that includes both screen positive and negative. So far, there's been 4 condition suspected results, and that kind of matches the sort of screen positive rate of about 1% that we were expecting. Of course, it's early days yet. We haven't had any condition suspected results yet in Central and South, although we're ready and waiting to receive them. It is worth mentioning that we've had 1 false negative result. That was for congenital hypothyroidism, and that's not unexpected. We're not expecting to pick up every case of CHT on the generation study, because many of those cases will not be genetic in origin, and that was picked up by a newborn blood spot screening. So it does just emphasize the importance that all our babies should have newborn blood spot screening, and that the generation study is not a replacement for that screening program. It's in addition to. Next slide, please. So just thinking about where we're going in the year ahead, we're still awaiting confirmation of funding going forward past March 25. We're fairly sure we're going to get it. It's a little bit nervous waiting for that confirmation, but that should be fine, and certainly continuing to work with our live sites going forward to support them where we can. Also, of course, working with our new sites to support implementation as soon as they are able. Like I say, we haven't had any condition suspected results so far, but we are ready and waiting, and of course we will need to monitor those pathways to ensure that our babies get the right support that they need if they find themselves in that situation. A big focus going forward will be on equality, diversity, inclusion, and preliminary work has already started on scoping our maternity populations to look at the ethnicity and deprivation within those cohorts, and also starting to compare uptake of the study with different ethnic groups. And we really need to consider how we can support diversity in recruitment going forward. So hopefully this time next year we'll be able to feedback on some of that work. And this course also links in to understanding why people decline, because it's important to get our messaging right about the study. last slide. Thank you. So that's all I was going to say on the study. If you've got any questions or if you want to hear more about it, then do contact me. I can come out to your teams and talk about it. Also, if you've got any ideas for sort of liaising with our sort of local communities, particularly in the West Midlands area, then again, do get in contact with me. Thank you.
Thanks very much, Karen. We're now going on to Sarah. Sarah, I believe you were wanting to present yourself. Is that correct?
Yeah, if I can just whiz through the slides that are perhaps a bit slightly less, not less relevant, but I just want to be able to whiz through.
While you're loading that, I was just going to say, if there are questions today, do feel free to pop them in the chat. And/or I think everybody will have put details on their presentations as well. So always happy to take questions directly as well.
Great, thank you. So I'm going to talk to you about the Severe Presentation of Infectious Disease Network of Excellence, which was one of the 8 networks of excellence that kicked off this year. I'm Sarah, I'm the Genomics Innovation Director, and I'm in a joint role between Health Innovation Wessex and Central and South GMSA. So as I said, I'm going to skip through some of these slides, but they'll be in the document for those who might want to go back and read them at your leisure. So just to remind you, the purpose of the network was to transform how patients with life-threatening infectious diseases are diagnosed and treated. And the great thing about this network is that it's looking at both the genetic information of the humans, but also of the pathogens, bacteria, viruses and fungi that are causing the diseases. And we'll be able to hopefully have a look and understand more at the end as to why some people have a more severe response to infection than others. This will lead to some targeted therapies and interventions for those patients, but also gives us an opportunity to provide some information to population health surveillance so that we can better respond in the future to situations such as the pandemic that we've just been through. So there's 3 work packages. You've heard me talk about this before potentially. The first one is respiratory metagenomics, the second is CNS in sterile sites, and the third one is host immunity profiling. So apologies, these are quite wordy, so as I said, I'm going to whiz through them. You can refer back to them if you're interested. So the respiratory metagenomics work package is led out of the South East GMS, and this is led by Jonathan Edgeworth and team, and this is looking at children and adults who are in a critical care environment with a respiratory illness, a respiratory infection. So GSTT have got a pathway that they've been working on for several years, and the purpose of work package 1 is to roll this out in a service evaluation model across other sites. The initial plan for phase 1 was to roll this out across 10 sites, and the purpose of it, or the benefit of it, is that it reduces the turnaround time of identifying those pathogens to 7 hours, which means previously where it may have taken 3 or 4 days to identify what was causing the infection and to start on treatment, patients can now start their treatment on the first day, which is incredible. So the first 10 sites, this is just a simple respiratory metagenomics workflow for those like me who have a simple mind. I found this quite helpful infographic, and this is the Guy's and St Thomas's slightly more professional looking one. And you can see here at the, oh, sorry, Vicky.
The slides aren't moving. I think you're suggesting that we should be.
Yeah, you definitely should be able to see.
That's this. We are now on the work package 1 leads.
Simple.
Go back to the simple one.
Oh my God. Okay. What can you see, Vicky?
We can see the work package 1, the word slides, the slide with bullets and words rather than graphics.
Okay. This is not good. Sorry.
That's all right. We're now on this.
So I'm going. Okay. Is it easier for Isabel to do this, and I'll talk to them.
We can certainly try. If you'd like
Apologies.
to jump back on. No, not at all. It's just one of those things, isn't it?
No, it seems to be a me thing, because I've always seemed to have issues with progressing the slides. I've obviously got a setting set up somewhere that is not working for me.
That's all right. Well, Isabel will pull them up and you can practice your next slide, please. Anyway, okay, good.
Okay, right. So yeah, this is the simple workflow. As I said, for my simple brain, this helps. And then Isabel, if you could go to the next slide, please. Yeah, so on this slide, what you can see is at the bioinformatic pipeline section, there's a little spin out that shoots the data off into MScape, which is the biosecurity element of this work package, collecting data there towards the pandemic preparedness type piece of work. So that's work package 1. The next slide, please. As I said, yeah, we've got 10 sites who are aligned to this work package. This winter, we've got 3 sites up and running, which is Great Ormond Street Hospital, Gurrs and St Thomas's, and UHS. This slide shows it's slightly out of date now, this slide, but this slide shows where the other sites are coming on board, and a huge amount of work being undertaken by the South East team to get us through this very kind of neat and tidy pathway. Lots of steps and governance, as you'd imagine. Next slide, please. The big news announcement that was a couple of weeks ago now is that £3 million funding to expand work package 1 has been agreed, which is super exciting. And that's in partnership with the Office of Life Sciences, the Genomics Unit, the UK Biobank and Oxford Nanopore Technologies. I think the funding is for about another 2 to 3 years. And the idea is to bring on another 30 sites. This says across England. Actually, that's incorrect. It's also including the devolved nations, which is great. Next slide, please. So just moving on to work package 2. Work package 2 is similar in as much as it's Judy Brewer in the UCL and GOSH teams have developed a pathway for metagenomics in sterile sites, particularly focusing on CNS. There is also some work in this space happening around bone and joint from the Oxford team, but for the purposes of this presentation, I'm just going to focus on the CNS. So Judy has got her pathway, and much like work package 1, she's looking at getting it to a state of preparedness where it can be rolled out across other sites. Her main focus is kind of exploring the pros and cons of the Oxford Nanopore technology and the Illumina pipeline, comparing the quality, speed, and the cost of that. Next slide, please. So Judy's successes this year are that she's had a UCAS accreditation of her protocol, which is really fantastic news. She's been able to recruit a new team member to support that piece of work, and her plan is to bring down the turnaround time to 36 hours, which massively increases the clinical utility of this tool, which is super exciting. Next slide, please. So yeah, moving on to work package 3. This is being led by Professor Julian Knight. This is looking at the human response, and so within this work package, the idea is that samples from both the CNS work and the respiratory work will feed into the host immunity pipeline that is being researched. This is much more at the research end of the innovation continuum, and some further sequencing of those samples would happen. Whilst the ethics and the consent process for that is being scoped, which is happening now, the team have been working really hard with setting up an MDT to look at existing cases, potential use cases. Could you go to the next slide, please? To take cases from the existing MDTs that are happening nationally, put them through this kind of potential overarching MDT to see what the benefit of host immunity may have brought to those patients. So if we go to the next slide, please. The MDT has shown, of the 7 cases that have been presented so far, that there has been a positive indication that recommendations could have been made for action in 71% of those cases that have been presented. So that's really encouraging. Next slide, please. And the data that we've started to gather, only of 7 patients, so it's it's kind of limited in its utility, but it kind of draws attention to the fact that we need to again consider the EDI implications of the work that we're progressing and making sure we're reaching all of our patients. If you could skip the next one and go to the what's gone well, please. Thank you. So as I said, yeah, onboarding of 10 sites is underway, with 3 sites actively processing samples. We've got expansion and funding for the next 2 to 3 years, and we're developing those relationships with O&T. And then, as I said, with Judy's work, she's showing a detection of suspected pathogen increasing 25 to 30% of cases, which the change in management of 75% of those cases. So the clinical utility of this work is already shining through. And then in work package 3, we've started to see the potential for next year, which is the feeding in of samples into the host immunity pipeline. So next slide, just wrapping up. Even better if is this that cross-pollination between work packages, some health economic evaluation across the network, building on the good data that we've already started, particularly considering equity of access, having a think about what's happening post 2026, and our SPID network event, which we had hoped to run in January, we have had a slight rethink, and we're going to offer a series of workshops, having a think about the complexity of introducing new diagnostic tests and including the data interaction and informatic pipeline development. Thank you very much.
Thank you, Sarah. Really helpful.
Thanks for your help.
No worries at all. So we're going to hand over to Steph Allen now, who's going to talk about developments in prenatal genomic testing. So Steph, over to you.
Hi, thanks very much. So I'm just going to give a very short overview of our developments in prenatal genomic testing. And this is on, although I'm presenting, this is on behalf of everyone who has contributed to this work. It's a huge team, both across all of Central and South and more widely nationally. So, and anybody who isn't involved, if you're interested and see things that you think you could link into as part of this talk, please do contact us to be involved in the future. Oh, next slide, please. I'm trying to do that on my computer. So really the backdrop and the whole background to this and the core of this is that at the start of the GLH, going back a few years, we consolidated testing to a single laboratory across the GLH, and this has been really important in the harmonisation of the testing and gaining experience and much larger numbers of samples. And you can see there numbers of amnios and CVS, so amnios blue, CVS orange, this has sort of enabled us to consolidate this testing. Next slide, please. And to support this, we've also developed a GLH and GMSA-wide prenatal clinical group with representatives from the laboratory in clinical genetics and fetal medicine, and we hope to expand that going forwards. And this is really sort of the core in helping us drive the service forwards in the future. Next slide, please. So in terms of, so we obviously deliver the core service and the routine service, but we're also now taking part in the prenatal network of excellence. So we're almost halfway through this at the moment, and this is collaborating with North Thames and nationally. And the number of work packages, I'm going to talk mostly about work packages 1 and 2 today. So work package 2 is really around the fetal anomaly pathway and invasive testing, so through amnio and CVS. Work package 1 is non-invasive testing, so further developing our testing of a blood sample during the pregnancy. Next slide, please. So first of all, the work package 2 in the fetal anomaly pathway. So this is focusing on fetal structural anomalies which are detected, and really extending that testing beyond the microarray testing through. We offer whole exome sequencing from October 2020, and it's really developing that further. Next slide, please. And it's important to focus that testing on anomalies which are more likely to have a monogenic cause. So really important that we work closely with our clinical colleagues. And this slide just shows the sorts of abnormalities that are associated with a monogenic cause, where we may offer R21 testing. Next slide, please. We carry this out by rapid trio prenatal exome sequencing. So it's sequencing the whole exome, but then analysis of a fetal anomaly panel of genes that are more likely to be associated with structural anomaly. Next slide, please. And there's a huge clinical impact of the prenatal exome testing. So this has been audited through the Express study and additional studies. So impacting during the pregnancy, delivery, neonatal management, future pregnancies. Next slide, please. And throughout the year, there have been a number of publications that we've inputted into talking about how this was implemented and factors associated with this, impact on outcomes, and how we might develop the service in the future. Next slide, please. In addition to that, we've been involved in a number of case and cohort studies. So, for example, we have eligibility criteria, and we need to, as more evidence comes into the literature associating different findings, structural findings, then and different things associated with monogenic disorders, then it's really important to get that out into the literature. So, for example, a number of papers published on new genotype-phenotype associations with disorders which had only been reported postnatally in the past, and then also auditing cohorts of patients. So, for example, the new criteria for isolated short long bones and small for gestational age, and what our pickup is with that, an isolated agenesis of the corpus callosum. So we're working across multidisciplines to audit those findings. Next slide, please. And then auditing our pickup of inborn errors of metabolism. So some of those are detected prenatally, and really important that we go back and audit which ones we've detected, and the sorts of scan findings associated with those. Next slide, please. But what are we still missing? So obviously we're not sequencing the genome, so we're missing anything else that's not within the exome. And also we're only doing our analysis using a fecal anomaly panel. So we're missing variants outside of genes outside the fecal anomaly panel. So really anything outside the exome, and other causes of disorder, so methylation, complex structural variants, and small CMVs. So although we're picking up the majority of things, what we need to now to progress our service further is really find what we're missing and try and improve on that. Next slide, please. So that's the aim of work package 2 of the Network of Excellence. So we're offering some gene agnostic reanalysis of our R21 data, continuing to do that in specific cases as requested by clinicians. We're continuing to explore progress towards whole genome sequencing. We're continuously reviewing the fecal anomaly panel because there's so many publications in the literature coming out. It's really important this is done regularly, and then reviewing the expansion of the eligibility criteria. And this is largely in collaboration with the North Thames group, who also offer R21. Next slide, please. So just as an example, the fecal anomaly gene panel, we review it every 6 months now, and this is becoming more routine. It's a huge undertaking. So the current review, we're reviewing 240 genes for inclusion, and that is really important to keep up to date so we don't miss diagnoses. Next slide, please. And then we're continuing to do gene agnostic reanalysis of our data as and when requested. So this is still ongoing, so we'll update on that further when we've done some more cases. Next slide, please. Sorry, next slide, that's it. So moving on to work package 1, which is around non-invasive testing. So we're all familiar with cell-free fetal DNA, hopefully, and the concept that you can take a blood sample during a pregnancy, isolate the plasma, and look for the cell-free DNA, and use that to predict whether a fetus is affected with specific disorders or specific conditions. And this is called non-invasive prenatal diagnosis. Next slide, please. So we've developed, prior to the Network of Excellence, a really good service, predominantly using a technology called RHDO, which is relative haplotype dosage. And we were the first really to use this nationally and internationally for a number of disorders. And to date, staggeringly, we've done testing for over 600 pregnancies across the UK. This is diagnostic testing, and you can see on the map down the bottom, because this is a blood sample which is stable during transport. We've had samples from Australia, Canada, Europe, so we're really widely taken up service, and we want to move this on further. So next slide, please. As part of the Network of Excellence, a lot of this is to improve our and develop further NIPD assays. So firstly, Lynn Chitty is leading on a national audit, and we're working with our data as well to identify any gaps, who's accessing NIPD nationally, whether there are any gaps where we could be offering non-invasive rather than invasive testing. We are developing new RHDO assays. So for example, we're working with North Thames. They've developed testing for the HBB gene, sickle cell anemia and beta thal, and this was submitted to the test directory. Another possibility on the horizon may be haemophilia, and we're looking at other possibilities. We're also extending availability of our RHDO testing, so now to consanguineous couples, and we've got a research project looking at long read sequencing, so Oxford Nanopore, to enable us to offer proband-free RHDO in the future, so no need for a proband to phase the high-risk haplotype. Next slide, please. And just to mention, as well as the non-invasive prenatal diagnosis for monogenic disorders, which is diagnostic, we also offer testing as part of the fetal anomaly screening pathway. So the NIPT, and Jenny alluded to the fact that we've tested over 15,000 samples to date. So we offer testing for 1/3 of England, and just notably the reprocurement for that is coming up in the 1st of April 2026. So I think we're thinking forwards in terms of what we can do with that service. Next slide, please. So in addition to obviously the testing for aneuploidy, it's really important that we explore it for other applications which may be applicable to the test directory rather than the screening pathway. So we've very successfully got a new indication within the test directory, so NIPT following a previous trisomy, that's happened within this year. We're also continuing to look at the possibility for translocation carriers, and also recurrent miscarriage, working with other teams across Central and South and nationally. Also, obviously, there's the potential to use this for subchromosomal anomalies and all chromosomes in the future. So this is something we would like to explore too. Next slide, please. So really, just as a wrap up, we're big developments so far, but we have an immediate future potential. So we really have to think further forward, because the NOEs were quite short time frames. So the finish in February 2026, which is not that far away. So I think we need to be thinking further forwards in terms of further funding. We've got massive cohorts of patients now because of our geographical footprint and the fact we're offering testing over a 1/3 to a half of England for some services. So really there is huge potential in terms of publications, patient recruitment if we wanted to develop further new technologies, so assessment of new technologies, and we have the potential to coordinate studies. So I guess this is just a plea that we can't do everything. We've got so much potential that if anybody else wants to get involved or input into this, then please do contact us, because so we can take this further forward in the future. Next slide, please. And then thinking even longer term, there are huge possibilities in the next 5 to 10 years, I would say, in terms of further development of non-invasive testing and how we might take that forward. And also, so for example, in utero fetal therapy is increasingly being presented in fetal medicine meetings, and I think that provides a real opportunity for us to develop further in this area, because obviously there's importance in diagnosis and particularly non-invasive within that space too. Next slide, please. So just a huge thank you to everybody. I can't name everybody individually because there's so many people involved, but across the lab and the clinical teams, referrers nationally, patients, women's and children's for supporting a lot of this, NHS England and FASP and the Network of Excellence wider team and our collaborators. Thank you very much.
Thank you very much, Steph. We're now going to hand over to Kate, who's going to talk about the Cancer Genomics Improvement Project. So Kate, over to you.
Thank you. Thanks, Isabel. Thanks, everybody. So just very, very few slides. This project is very much in its infancy, but I think it's useful to kind of set out the scene for those of you who don't know what we're doing. So the Cancer Genomics Improvement Project, or CGIP, is a project that's initially funded by NHS England for 6 months. At the moment, it comprises me as the quality improvement lead across the geography, and Kat Omar, some of you will know, who's the project manager. So as I say, at the moment, we're focusing on 6 months funding with the view that we're hoping that it will be extended beyond that. So next slide, please. Okay, so I've got a couple of these slides, which I've mercilessly kind of used from NHSE previous presentations, so some of you will have seen these, but I think this slide and the following one, this one is around solid tumours. It just kind of sets out at a very high level the complexity of the pathways around genomic testing for our cancer patients. So I don't wish you to look at the detail necessarily, but I think one of the things that's useful to take from this slide is that it's complicated, and that there is a need to kind of Isabel, have we missed a slide? I think we've missed a slide. Sorry.
Oh, apologies. Not to my knowledge, but I can go back and look in.
Doesn't matter. I could talk around it.
Thank
It
you.
doesn't matter. The previous slide I thought was coming was the reasoning, the rationale behind why we're doing CGIP. So let's take a step back. There's a need to look at the end-to-end pathways in this space, because there are some new nationally set out turnaround times which need to be implemented over the next sort of year or so. And they are very much kind of set out specifically for solid tumours and for hemato-oncology, and they are subtly different, but they are going to be a challenge for us to deliver. So the CGIP project is focusing on how we achieve that. And you can see there are some arrows in there that sort of denote the times, if you like, that we are expecting that these kind of parts of the process will be squeezed down to. What I wanted to show you from this slide was the complexity of the pathways, but also just how bound we are on kind of the lack of integration, I guess, in the informatics systems that we use and the different laboratory LIMS systems that we have in place. And every time you see a red square there and a box around those pathways, you can see that there is a need to manually enter data. So one of the things that we're looking at is how we can work around that and make improvements in this space, allowing for the fact that there is a lack of laboratory integration. So this is the solid tumour pathway. There's the clinical part from where the specimen is taken. That's when the clock starts ticking. The sort of green bit in the middle, I guess, is what happens in the histopathology setting. And within that, you'll see there's a purple box that straddles that and the GLH, which is around the CPGCs, which I'll talk a little bit more about in a minute. And those are newly created to kind of smooth the transition, as it were, in this space between histopathology and the genomics laboratory. And then the report is generated from the laboratory and goes back to the clinician, and that's when the clock stops ticking. So if I could have my next slide, I'm hoping the next one will show you a similar schematic. There we go. For hemato-oncology, again, same idea. The red boxes denote the manual booking in. The little buses on there, I guess little vans, are where we have to physically move the specimens around the region. And for our region, geographically, that is quite a challenge. So if I can have my next slide, please. Yeah, so this is the last slide that I've got to show you, but just to say that there is an action plan that's been drawn up and agreed with the genomics unit for us in the Central and South region. There is one of these kind of action plans that exists across the whole of, across the 7 GLHs in England. Their kind of aims, if you like, are similar, but they have different challenges. And these are the 3 initial actions that have been agreed for us in Central and South. So the first of those is to establish and embed reliable transport from our newly created CPGCs, cellular pathology genomic centres, and those kind of transport links are absolutely key to getting the specimens moved around the region effectively. There are currently 3 main sites: Wolverhampton, which is very much established, Southampton, which is still evolving, and Oxford. And there's a 4th one of those, which has yet to be confirmed, but hopefully towards the end of January 25, we'll know where that 4th centre will be. And then the second action sounds so simplistic when you see it written down here, but actually it's everything to do with the pre-processing of our specimens for everything from a sample receipt all the way through to the end of DNA QC. The moment we're focusing on that initial sample receipt triage. I've been to Southampton and Salisbury and looked at how they do it there. Kat and I are going to Oxford in January, so we'll be able to look at what happens there. And we've done quite a lot of kind of witness audits, that type of thing, in the Birmingham laboratory too. The second part of that, we're breaking it down, is around the DNA extraction processes, and there is a piece of work already ongoing around harmonisation of how we do that, all of the things that contribute to that, with the view that if we can make some changes in this space, we can reduce turnaround time. There's also a large review going on around the FFPE pathway in Birmingham, and how we do that, and whether there's some changes there we can make. And then the third and final one for this initial 6 months is around reducing the time taken to perform the analysis and reporting. And there's a separate working group looking at that. Lots of ideas on the table, again, working across the whole region to learn from good practice, learn from each other, and to see what we can do to shave off some time. So I think that's me. I think that's the end of my slides.
Yep, that's everything. Thank you so much.
No problem. Thank you.
So just moving forward, we're going to hear from Tony Williams and Carolyn Campbell, who are just kind of wrap up with what we're looking forward to for 2025. So Carolyn, I think it's over to you first for the GLH.
Thank you, Isabel. So yeah, I'm just going to very quickly give a brief overview of where we think our major focus will be for the coming year. So you've heard through some of the talks already that new target turnaround times have been introduced across multiple pathways this year: rare disease, solid cancer and hemato-oncology. And while we're already working towards these based on trajectories that the Genomics unit and our own GLH have developed, we are expected to implement these fully from April 2025. And we will work alongside Kat and Kate with the CGIP programme to help achieve that reduced end-to-end turnaround time for cancer patients. And we're also constantly reviewing our workforce and our skill mix and how we use our workforce optimally in ways that will allow us to enhance our efficiency and release capacity. Most of you, I think, will be aware of the publication of the ACGS position statement this year. And so early in the new year, the GLH will be standing up a working group to evaluate how we could implement single sign-off across some of our pathways. And we'll continue the work that's ongoing across all of our laboratories, looking at ways to introduce automation into our reporting processes. You'll also probably all be aware that our current contract for the GLH ends in March 2026. And although we do not yet have a fully confirmed timetable for what might happen over the next 15 months, we're certainly expecting market engagement for the new contracting process to commence early in the new year. And we're expecting an invitation to tender probably from the beginning of 25-26. Moving on then to training and education. Our staff are very clearly our most precious resource, and all the things that Jenny talked about that we have achieved this year will obviously continue in future years to ensure that we have the appropriate workforce for the future service that we need to deliver. With the introduction of different models of service delivery across the GLH, we've had to adapt how training is delivered specifically for those on the STP programme. And our STPs now receive training in all of our labs across the GLH, and they rotate around different labs for different aspects of that training. And that's been a really positive advance, I think. The next step for us is to consider a GLH-wide accreditation for our training rather than each of our labs being accredited separately. And that's something that we're going to be taking forward early in the new year and trying to learn from the work that's already been done in the North Eastern Yorkshire GLH, where they have this year achieved that consortium-wide training accreditation. You've already heard quite a lot about Central and South involvement in the prenatal and SPID networks of excellence. Central and South also have a key role in 2 work packages of the hemato-oncology network of excellence, and that's work that will be undertaken primarily in the Oxford labs. One of those work packages is looking at developing an NGS-based assay for myeloma, and the second is looking at developing a rapid 7-day turnaround time for whole genome sequencing in hemato-oncology, with a view to being able to replace a lot of the current standard of care testing and the WGS becoming the standard of care test for acute leukemia. In pharmacogenomics, we know that we can expect the number of different pharmacogenomic tests and the volume of these tests to increase very significantly in the future. So the GLH is looking at the best ways to deliver these tests, both in terms of technology and also in terms of what infrastructure will be required to deliver this very high throughput testing. And then finally, we will obviously continue to be responsive to the many and varied other changes and challenges which are undoubtedly coming our way over the next 12 months. And then all that's left for me to do is to sort of reiterate my thanks for the work that has happened over the last year, and to thank everybody in advance for the huge amount of work that is going to happen again in the next 12 months.
Lovely, thank you, Carolyn. So I'm just going to hand over to you, Tony. Just click through.
So thanks, Isabel, and thanks all. My name's Tony Williams, the Clinical Director of the GMSA, and it's fantastic just to hear everything that's just been discussed over the last 50 minutes or so on just some of the achievements and the way in which things have moved forward year on year within the programme. I guess when I was reflecting on it, my big reflection from this year is sort of the power of people and how everybody here has been working individually and collectively and dedicating their time and expertise. And it's that dedication actually that's the core of what is achieved against whatever is put in front of us. And that's a concept of being better through partnership, hopefully will stand us in good stead as we move forward into next year. And as alluded to, the big challenge next year, over all other challenges that we have with the government projects and plans in place, is how we generate our next step to what happens beyond the current cycle of funding. As we've seen, the emergence and evolution of the service is continually in flux, and we need to be working in harmony to provide the best strategy going forward, our advice to the teams working above us, and allow that to come together on behalf of what we see as going to be the best way of delivering care to the patients. And the bottom statement there is that we don't have to have it all worked out before we move forward. And I think we'll have to have that with us over the next year, because things won't be as clear as we might like. But whatever they may be, collectively, we will be able to work through them and with them and make progress. And in terms of the partnerships and the sort of visual icon there of how we work together, we have really focused, I think, on many aspects in the last year, starting with our executive teams. So for those who cast their minds back to January of this year, we brought all the executive leadership team, the chief executives of the trusts that have supported us to the greatest effect over the last few years, the senior teams that they work with, to get that buy-in at our trust level so that we can begin to deliver some of the objectives. And then the core clinical teams, the lab clinical teams, the core clinical communities across rare disease cancer, the pharmacogenomics program, emerging infection and public health. And then our institutions, those which we partnered with and evolved stronger bonds, our cancer alliances, our pathology networks, our additional colleagues that we brought in from different sectors. Then the academic communities that we've held with, our group that we do a symposia with, that bring in to help us and support and contribute to the developments we take in aspects of delivery of the lab side and the clinical side and publications, and then the investment that they bring. And I guess then lastly, but also ever so more important, our commercial partnerships that we are beginning to substantiate so that they can give us strength going forward. And then just a few sort of highlights then in terms of where I think these foundations from this year will take us forward. And the principal partnership is with the patients and the public. And so pleased to see the launch yesterday of the Raised Health Observatory programme that we've been involved with, and a test into what we'll be able to achieve in the space of that programme, and working with some of the excellent work our teams have done through Tony and colleagues with the patient and public voice. And then the cancer community, I think the CGIP has been mentioned, but also the Cancer Reference Group and the way which has been able to bring our large geography and sometimes a more complex picture than other areas together to deliver change that is equal and equitable across the whole region. Those research partnerships that we've already commented on, either those external to us, as it were, with partners such as our Genomics England colleagues leading the generation study, or some of the other ones that some on the call have led independently and collaboratively with other members of the CAS community and outside of the CAS GMSA. The innovation program, again, has begun to evolve, and again, the connection there with not only the programs within scope, but what else is going on across our health innovation networks in the West Midlands, Thames Valley, and in Wessex, and trying to integrate the networks you've already been described around, as well as the other ones we have in the artificial intelligence network of excellence and the programs within the rare disease program. So we've got at least 5 programs that we're actively involved with. And then just to conclude then, the ever-increasing professional groups that are now both multi and interdisciplinary that come together to give us this richness, which is the GMS going forward. And as you can see there, there's many groups, but particularly groups there that have come together in this last year to deliver some of these partnerships, which I think will now be the foundations of how we lead them into delivery vehicles in 2025, and through them, as I say, our model for what we'll become in 2026 onwards. So again, a big thank you for everything people do, but particularly the time and commitment. There's so many pressures on time commitment, and sometimes many will go over and above, and it's appreciated, but also hopefully everyone will take some rest and a little bit of leave over the next couple of weeks or so. So big thank you.
Thank you so much, Tony. We've now got a bit of time for questions. I'm sharing, so I can't actually see. So Vicky, if I could hand over to you to see if anybody's put anything in the chat or puts their hand up.
Will do. And I think there's Steph and Jenny have had a bit of a sort of think about potential for methylation work to expand prenatal testing, and then from Jenny, and then a response from Steph. I'm not going to pretend to do justice to that. What I am going to say is that I think really useful information there, and also perhaps the opportunity to do a focus webinar on prenatal in the coming year. And I would just say to everybody on the call, we hope this has given you a flavour. We hope this has said thank you as well. And I know we can't cover everything, but trying to get a sort of rounded view of what is going on. We'll aim to hold this annually, so dates will go in diaries. And as I say, if there's anything you would like to put forward to go into the document for the review of the year, do let us know by the middle of the week. Never want to miss an opportunity to get a plug in for something else. We have our face-to-face event this year on the 1st of May, and that is in Southampton this year. So we're down at the football club in Southampton. So if you've got very strong allegiances, I don't know what that does, but we're just using it as a conferencing venue. So that is the QR code. We'd love to see you there. I know it's a challenge to sort of get time away from desks or benches or clinics, but if you can make it, it is a great opportunity to come together, and seeing people face to face just adds richness to those conversations and how we push on for what is going to be a sort of really important year. So I will close us off there if there is nothing pressing. I'd like to again reiterate thanks, but thank you to the speakers today. Really appreciate it. I always learn something new just from those 10 minutes sort of pen picks of what's going on. So a huge thank you and a very happy Christmas to everyone on the call. As Tony says, I hope you get a little bit of downtime, and really looking forward to working with you in 2025.